Hello, my name is Erwin Datema. I'm a senior scientist at KeyGene, and today I would like to talk about ultra-fast comparative genomics of complex genomes at population scale. So I work at KeyGene, the crop innovation company, and our ambition is to enable breeding companies to improve their breeding efficiency, to obtain unique varieties that answer the needs of growers and consumers, and to support our customers in answering their toughest research questions. We achieve this using state-of-the-art DNA-based knowledge and technologies, and we work on ornamentals, field crops, vegetables, industrial and food crops. So I work on a lot of plant genomes, and plant genomes range a lot in size, starting from very small genomes such as melon, only a few hundred megabases, all the way up to large genomes like wheat, 17 gigabases. And just as a point of reference, the human genome at its 3 gigabases is only about a quarter of these large genome size. Not only that, but plant genomes are also very complex. They can be repetitive, they can be really heterozygous, and they can be polyploid. For example, red wheat is a hexaploid, potato is a tetraploid. And the current tools that we have available to look at these genomes, to assemble them, but also to compare them, focus primarily on simple human genetics, so diploid genomes. Well, in the plant field, we're entering the pan-genome era. What we have seen over the last couple of years is that for many plant species, there is a substantial amount of genes and other interesting genetic variation that is not present in the reference genomes. This is found by sequencing many different accessions and taking, for example, a look at the genes which are present and absent, as has been done in maize, or looking at genes that are unique to subsets of accessions, as has been done in brassica, or to just take reads, map them to a reference genome, and find out that there's a lot of pieces missing from the reference genome. And all these pieces that are unique to these genomes actually contain relevant information that we want to use during breeding. So what we started in, let's say, about 8 years ago was a concept called multi-variome analysis, where we take the reference genome that we have available, we know the annotation of the genome, we then map short reads of a certain accession to this reference genome, take another accession, also map the short reads of this accession to the genome, and then we can start identifying variants, so small SNPs and indels. And actually we can look at copy number variations in genes, so to identify which genes occur in which individuals and which genes may be present in multiple copies in some individuals. For about 4 years later, we started looking at this in a different way. So we took the reference genome, again, mapped the short reads from a certain accession to it, but also had a look at the unmapped reads. We took those unmapped reads, assembled them de novo, and then added them to what we call a pan-genome. And then we took the second accession, mapped the short reads of the second accession to the pan-genome that we had. Again, took the unmapped reads, assembled them de novo, and extended our pan-genome. And in this way, we could then annotate the extended pan-genome and identify, for example, genes that are present only in subsets of accessions that are not present in the reference genome. And of course, we can also do all the normal analysis, such as SNP mining, finding structural variants, etc. Now, with the advent of long-read sequencing technologies, such as HiFi sequencing, we can actually sequence complete genomes de novo and also multiple genomes from a single species. So now we can take the reference genome for one accession and compare that to the genome of another accession. But of course, we don't only have 2 accessions, we have multiple accessions. So we can build what we call a pan-genome from this by comparing all these genomes to each other on the whole genome level. And so we identify blocks of locally collinear regions, so that are regions that are present in a subset of the genome and that are collinear with each other, and then we can connect these blocks into a graph. And then each path in the graph represents a original input genome. So each of those genomes may have a different order and possibly orientation of those blocks, and may also contain several blocks that the other genome doesn't contain. And based on this, we can then again provide an annotation and look at genes that are present and absent from individual genomes, for example. Now, in order to achieve this, we need a comprehensive computational toolset, starting from de novo genome assembly and ending all the way at interrogating and visualizing the data that we get from these genomes. In my talk today, I want to focus on the right-hand side of this picture. So starting from pairwise whole genome alignment and then building a pan-genome out of these multiple genome alignments. So plant genomes, as I told you before, are quite challenging. They are very repetitive, and by repeat, I mean, for example, the retrotransposable elements, but also gene copies that may occur in large clusters spread throughout the genome. And so if I take two genomes and I want to compare them to each other, I can draw what we call a dot plot, where we draw a dot wherever two genomes are the same, and these dots end up connecting into lines. Now, if I have a genome where there is one certain element that is shared between the two genomes, I will get a small line like this. However, if there's two copies of the same element in the genome, I end up with 4 alignments. 3 copies, you can guess, 9 alignments. And I can extend this to many copies, and in the end, we get many squared alignments. So we get all these little pieces of sequence that are the same. And this, as I said, this happens a lot in plant genomes. But how do we get a best collinear path, so a best whole genome alignment from all these bits and pieces that are the same? Well, this is a very large computational bottleneck. And so in order to solve this, we started looking at this from a different way. Can we reduce this complexity? And of course, at KeyGene, we have a very long history of complexity reduction and actually looking at restriction sites. So what we now do is we take the two genomes that I call here the subject and the query genome, and we make an inventory of all the restriction site motifs on these genomes. We flank these by short sequence tags so that every motif gets to some extent unique. And then we know the distance between every consecutive pair of motifs. And using this information, we can now align these two genomes on a very global level. And as you can see immediately from the image, this also means that every subsection of the genome that is aligned now becomes an independent and therefore parallelizable problem. So, of course, we did not invent all of this by ourselves. I mean, restriction map alignment has been around already in the 80s. In the landmark paper from Waterman, Smith, and Ketcher. Also, finding motifs in a sequence is basically the same as finding words in the text. And this is also something that has been done already for almost 50 years. Comparing these short sequence tags is kind of similar problem to a spell checker, where you just need to find a small number of differences between short words. And finally, to align the sequence in between the restriction sites, we make clever use of sequence alignment heuristics, such as those found, for example, in the FASTA algorithm. Well, taking these algorithms together and combining this with the innovations of 30 years of KeyGene, we have now built a super fast and highly sensitive genome aligner. Of course, this doesn't scale only to the problem of two genomes. We can also scale this to progressive multiple genome alignment. So if we want to build a pan-genome, we have to compare tens or maybe hundreds of difference of genomes. So starting from a pairwise alignment of two genomes, we can now collapse this in a sort of consensus, and then take this consensus and align the third genome to it. Again, using only these restriction motifs and short sequence tags to identify where the genome should align. And actually, because we can make these pairwise alignments very rapidly, we can also build a phylogenetic tree to guide this process so that we are sure that we align the genomes in an order that gives the highest sensitivity and specificity. Now, when I talk about fast, I mean really fast. So to give an example here, we can compare two melon genomes, which are about 400 megabases in size, in about 13 seconds. And this is using a single machine, a machine that is commonly found in whatever bioinformatics group is closest to you. Tomato genomes, which are about a gigabase in size, take less than a minute, and even relatively large genomes already, like lettuce, take less than 2 minutes for complete alignment on the nucleotide level of two genomes. Now, this idea scales to any problem. Large genomes, very repetitive genomes, such as pepper, can be aligned in about 5 minutes. Another example is hexaploid breadwheat versus tetraploid germ wheat. These two species share two genomes, and the hexaploid genome has an additional genome. In order to compare these, it only takes less than 15 minutes. So this is about a coffee break in order to get a full nucleotide level alignment of these two genomes. And many of you are maybe not so familiar with plant genomes. So I also tried the human genome, and it took about 1.5 minutes to compare two human genomes that I dug up somewhere from the public domain to compare these at the nucleotide level. Now, of course, it's nice that this works, but what is the precision? This is, of course, very difficult to answer for whole genome alignments, because often we do not know the ground truth on what base should align to what other base exactly. So what we did there is we had a look at different public tools that we have used in the past and compared the output of our aligner to the output of those aligners. And what we saw is that for more than 90% of the bases, they were aligned in an identical fashion, also between the 3 different tools. That's what is not shown here. So taking this together, we can now, in a matter of seconds or sometimes minutes, align very large complex genomes at a very high accuracy. And keep in mind, other tools easily take hours or days for this. Now, pairwise alignment is nice, but when we want to build a pan-genome, we need to align multiple genomes. So how does this technology that we developed scale to multiple genomes? To answer that, we had a look at 3 public RISE genomes, again, relatively small genomes, and we took some tools from the public domain that are often used, like seq-seq-pan or progressive MOVE, to build a pan-genome. With seq-seq-pan, it took about 6 hours, which is quite reasonable if you want to build a pan-genome from 3 genomes. Progressive MOVE finished in an hour, which is nice. But our own tool that we have named STL Progressive right now, STL stands for Sequence Tag Layout, the technology that is based on the restriction sites, the short sequence tags, and the layout of those to make a very rapid alignment, only took 1 minute. Now, how does this scale to large number of genomes? To answer that, we took 9 plant chromosomes. The chromosomes were about 100 megabytes in size. We had 9 different individuals. We took the same chromosome, and we again asked these 3 tools, can you make a multiple alignment of this? We gave them a week. seq-seq-pan did not finish. Progressive MOVE finished in about 4 days. And our own STL Progressive aligner finished in 2 minutes. Now, of course, single chromosomes is nice, but what about real genomes? Well, this single chromosome, of course, comes from a real genome. And we actually took the entire genome, which is a 4 gigabase tetraploid plant genome. And again, we gave it to all the 3 tools. Well, seq-seq-pan and Progressive MOVE could not make the alignment in 1 week, whereas STL Progressive took slightly more than 1 hour to finish the complete alignment of these entire genomes. And so this technology now allows us to build pan-genomes at population scale, because this scales almost linearly. So what we also need is, of course, a way to interrogate these data. And as you know, in the linear genome world, we're used to working with FASTA files. And we can run BLAST against a FASTA file or GeneFinder against a FASTA file. But now in a pan-genome world, we need to think about this differently. So starting from the progressive multiple sequence alignment, we now extract the locally collinear blocks. So these are the parts of the genomes that are similar between them. And we build this pan-genome graph, as I've shown before. And now we take this pan-genome graph and the input genomes and store them in specialized data structures that we connect through APIs to what we call our pan-genome store. And this pan-genome store is then connected to the outside world to a RESTful API so that external applications, such as analysis tools and visualization tools, can query this pan-genome store, for example, for extracting a certain region from a certain genome and all the pieces aligned to it, or a certain gene and all the pieces aligned to this. And we can very quickly now extract specifically the information that we need from such a large pan-genome graph and visualize it or interrogate it. Now, what I have tried to show you today is that we have entered the pan-genome era. And so we are shifting away from the reference-centric view of a single genome towards a collection of genomes that contains all the nucleotide and structural information that is present in a population. And this is possible because genome assemblies and also sequence comparisons, as I've shown today, have become commodities. They can be executed relatively fast, and they don't cost a great deal of resources anymore. KeyGene has the tools to analyze these data and tools that scale with increasing data volume. So aligning 9 complex polyploid genomes only takes an hour. And of course, we are now rethinking the downstream sequence analysis tools that we need in the pan-genome world. Like I said, it's easy to blast a sequence against a linear FASTA file. But how do you blast a sequence against the pan-genome? Well, this is the topic that we're working on today. What I also wanted to show you is that many of the algorithmic problems that we face actually were solved decades ago. And at KeyGene, we are inspired by knowledge from these other domains. For example, natural language processing. And we're trying to learn from this as much as possible by taking a look back at all papers, at all technologies that may have already solved the problems that we are facing today from another angle. We like to combine these with suitable heuristics. So heuristics that make very challenging problems suddenly a lot simpler and that make what was a couple of years ago impossible to achieve, a pan-genome from 9 polyploid large genomes, to make that easy, to make it achievable within an hour. So with that, I would like to end my presentation, and I would like to give you the opportunity to ask me questions, either during the Q&A session or just by contacting me or KeyGene directly. Thank you for your time.